Recording.
All right, great. So thanks everybody. Welcome. I'm Stephen McCord, part of the Columns Leadership, NorCal Director. That's my current title. And this is the first webinar for the year or this season. We've got about 5 or 6 lined up through about monthly through June. And Austin's got it first. But before we get started with that, as noted, just some housekeeping stuff. Please mute your phone so you don't inadvertently cough or burp or make other sounds into the audio to interfere with things. And before I introduce Austin, let me give our sponsor a moment. Lisa's here, and I'll pull up the right slide. Oh, screen. There you go. All right, take it, Lisa. Oh, maybe you're on mute too.
Yeah, I just unmuted. Hi.
There you go.
Everyone. I just wanted to take a second to talk to you about EcoRx. We've had success with this over in New Zealand. It's a wave-powered gravity pump that takes oxygenated water from the surface and kind of pushes it down into the water, which basically prevents algae blooms from forming. So we just started manufacturing these in Anthem, Arizona. This is one example. We have different prototypes we're kind of working on now, and it really just helps to mitigate low oxygen levels in the water, in lakes, ponds, estuaries. We can, with one pump, basically take care of about a football field size on a lake or pond. And we're looking at production costs of around, $6,000 to $8,000 per unit. So we're pretty excited, and this is the work that we've done in New Zealand, kind of what they look like, and we've had some success with that. So that's really just kind of all I wanted to talk about. I'll keep it pretty brief. I'll put a link to our website in the chat, and we'd love to talk to you guys more about it. Thank you.
All right, thanks. I'll just slip through those. So there's the link, but you'll post it in the chat.
I'll post it in the chat too. Yeah.
Great.
So thank you very much.
Sure, thank you. And before I turn it over to Austin, just a quick introduction. He's a research hydrologist with USGS Idaho Water Science Center in Boise, Idaho. Calling in from there. His research focuses on field-based studies of contaminant sources, transport, fate, and potential biological effects to surface waters and sediments. And this is a project I heard about last year in Hell's Canyon. Actually, I first heard about it in the 90s because this area has been researched for many years. But Austin can share some really interesting results that they've had from a wealth of studies. So go ahead and take it from there, Austin.
Okay, thank you, Stephen. Let me share. Okay, I think you're seeing me now. Are you seeing your own little window down here in the bottom corner?
No.
I don't know if I can. Oh, okay, good. Okay, yeah, thank you again for having me, and thanks everybody for tuning in. Like you said, I'm with the USGS, and this is a study that USGS has been working on with a number of partners: Idaho Power, Portland State, UC Davis, Idaho Department of Environmental Quality, Reed Harris Environmental, and University of Wisconsin at Madison. So this talk is titled, and Stephen helped me with this title, Come Hell's Canyon or High Water, which was great, Stephen. And this is about methylmercury production in Hell's Canyon, which is on the Idaho-Oregon border. Just in case you thought that this talk was about the Deep Purple album, Come Hell or High Water, that's a different talk. And as is the talk, the romance novel Come Hell or High Water, featuring the tall and handsome Cade Silver. That's a different talk as well. This talk is about mercury in Hell's Canyon, Idaho-Oregon. So let's go there. Hell's Canyon is a series of 3 reservoirs on the Idaho-Oregon border. This overview map, let me get my pointer going here. This overview map shows the Snake River flowing through the southern part of Idaho, east to west, and then it hits the Oregon border and flows north. this box shows the area of these 3 reservoirs. Downstream of here, the Snake River flows into the Columbia River, into the ocean. Here's a satellite image of what these things look like. So the first reservoir is Brownlee Reservoir here, then Oxbow Reservoir downstream, and then Hell's Canyon Reservoir downstream of that. This longitudinal schematic here in the lower left shows how long and deep these are. Brownlee Reservoir is the longest and deepest, about 55 or 60 miles long and about 270 feet deep, which is about as tall as the Statue of Liberty. There's not actually a Statue of Liberty submerged in Brownlee. Oxbow is the smallest, and then Hell's Canyon is kind of in the middle. Most of my talk is focused on Brownlee Reservoir because that's where we've kind of learned over the years. That's where most of the action is in terms of mercury. And last point, together these 3 dams generate about 30% of all the electricity that Idaho Power produces. Here's just a couple of pictures of Brownlee Reservoir, just to get you a sense of what the area looks like. So here's a little boat down here for scale, very arid environment. Here's a couple pictures of Hell's Canyon Reservoir. This is Hell's Canyon Dam. Again, for scale, this dam is about, I think, 270 feet tall. So this is a pretty big canyon. And yeah, pretty steep canyon walls. Upstream of the Hell's Canyon complex, the Snake River flows through the Snake River Plain across southern Idaho, which is a semi-arid desert, which with about so for example, Boise gets about 13 inches of rain a year. So all of this green that you see along the Snake River corridor here, this is mostly not natural green. The hydrology in this area is dominated by mountain snowmelt. So here's some mountains here. That is natural green, but a pretty elaborate system of dams and reservoirs and canals capture and divert that irrigation, that snowmelt water, to irrigate farm fields along the Snake River. So if you zoom into this, you see a lot of farm fields. Some of them are center pivots. A lot of them are flood irrigation. So those of you in California can appreciate, I think, the scale of canal building here. This has been likened to an irrigated Eden by this book here. Sugar beets, alfalfa, corn, and of course potatoes and other crops. There's large-scale dairy operations, including the largest yogurt factory in the world by Chobani, fish farms, cattle feedlots, etc. So as you can imagine, with all this agricultural abundance, there are some negative side effects to water quality. Some have likened the Snake River to a working river. Others have called it Idaho's sewer system, as High Country News did in 2014. And that's because if you float or boat down the Snake River in the irrigation season, it's not uncommon to see direct discharge of irrigation water right into the Snake River, and that water is pretty chock full of sediment and nutrients and anything else that's coming off of those fields. So both upstream and through the Hell's Canyon complex, the Snake River is impaired for a variety of water quality constituents, including nutrients, sediment, algae, and water temperature. So how does all this relate to mercury? Real quick, here's a little diagram of the mercury cycle for those of you who may not work on mercury topics. Mercury comes from a variety of natural and anthropogenic sources, but coal-fired power plants are a big one, emitting mercury into the atmosphere and then that mercury deposits on the landscape. Some of that runs off into the nearby wetland or stream or lake. And then in the right conditions, that mercury can be converted to methylmercury by anaerobic bacteria and archaea. Methylmercury is the kind that we don't want. It's a potent neurotoxin, and it bioaccumulates in organisms and biomagnifies up the food web. Methylmercury is a problem nationwide. A study by USGS found or estimated that methylmercury concentrations in fish exceed the EPA human health criterion of 0.3 parts per million at about 1 in 4 streams across the US. And methylmercury contamination accounts for 81% of all fish consumption advisories in the US, which exist in every state in the US. And methylmercury in fish is especially problematic in reservoirs. This is a figure from Williker et al. 2016, comparing fish mercury concentrations in lakes, shown in blue, versus in reservoirs, shown in red, in different ecoregions of Western North America. And you can see that reservoir fish concentrations are about 1.5 times those of lakes. Okay, so that brings us to the subject of today's talk, which is mercury in the Hells Canyon complex. Several years ago, it was observed that some of the fish in the Hells Canyon complex exceeded this human health criterion of 0.3 parts per million. So in 2013, the USGS, Idaho Power, and Idaho DEQ started this 10-year study to understand mercury sources, transformation, and biological uptake in the Hells Canyon complex. The objectives of this study are to identify the drivers of methylmercury production and uptake into the food web in the Hells Canyon complex. Also to inform a C== W2 mercury predictive model to test management scenarios, which can then be used to develop a mercury management plan, and to provide data and guidance for the development of a Snake River methylmercury TMDL, which the state of Idaho has recently started working on. Okay, I don't have time to present everything from our 9-year study, of course, so I'm focusing on a few key topics or key findings in this talk. And these are pretty much pulled from 4 different publications that either came out in the past year, and that's a publication that I led and a publication that Jesse Namick, who's on this call, led, and then 2 publications that are coming out in the next year, later in 2013, and those are led by Brett Poulin and Ben Peterson at UC Davis. Okay, so the first finding here is that excess nutrients and organic material from upstream drive seasonal anoxia in Brownlee Reservoir. All right, so we're going to watch a movie. This is an animation of temperature and dissolved oxygen in Brownlee Reservoir. Temperature on the left, dissolved oxygen on the right. So this is a longitudinal cross section of Brownlee Reservoir. So flow is coming in from the right, and the dam would be on the left. If you can't see the scale bars, which you probably can't, on the temperature side, blue is cold water, like 4 or 5 degrees, and red is warm water at 24, 25 degrees. On the dissolved oxygen plot, red represents anoxic water at a dissolved oxygen of less than 1 or 1, and blue represents oxygenated water at 14 or 15 milligrams per liter. So these profiles that I'm going to show you were created by vertical casts of dissolved oxygen and temperature sensors every 2 miles up the reservoir. This is what these dotted lines represent. Every 2 miles up the reservoir, vertical casts to collect these data. And those are done every 2 weeks by Idaho Power, by Nick, who's on this call. And then they're kind of stitched together into a movie. So we're gonna watch. I had to get my laser pointer off because laser pointer doesn't work. Okay, so we'll watch this movie and I'll stop it a couple times to make a couple comments. We're going to start in March of 2018 and we're going to go all the way to November of 2018. So notice that in March, the whole reservoir is pretty well oxygenated and fairly well mixed in terms of temperature. We'll watch as it stratifies and becomes anoxic during the summer, and then as it destratifies and becomes well mixed again in the late fall. So keep an eye on the dissolved oxygen plot. About midway through the reservoir, we start to develop some anoxic conditions in May. You see that orange that's starting to spread and turn into red. And those anoxic conditions spread down towards the dam, but also up reservoir into the transition zone. All right, I'm going to pause it here for a minute. So now we're in late July. We have anoxic conditions throughout a large part of the reservoir from the transition zone up here at river mile 315, 320, all the way down to the dam. We also have, on the temperature side, we have pretty good stratification of the water with really nice warm water up at the top and cold water down at the bottom. So as we move into August, when I restart the video, keep an eye on the dissolved oxygen plot in that transition zone, that anoxic water in the transition zone, river mile 315, 320, 325. We're going to have cold water start to flow into the reservoir in August, and it's going to plunge beneath the surface, and it's going to start to erode away and mix in these anoxic layers. All right, here we go. Okay, so there went a bunch of our anoxic water mixed in and sent downstream. Now we're into September. Oh shoot, I just restarted on accident. Let me fast forward it. Okay, back in September here. Pausing it again, just to show you that you can see these lobes or tongues of cold inflowing water plunging, interflowing into the reservoir. I should also point out that the penstock or the outflow of the reservoir is around an elevation of 1950, and you can see how that affects the structure of the temperature and dissolved oxygen. So colder and colder water coming in, plunging deeper and deeper until very little of that anoxic water is left. Okay, so that's where the movie ends. That's late November. Nick tried to get a profile in December, but his sensors froze up and he wasn't able to get those data. But I think in December we would have seen that little remnant part of anoxic water there that we see in this image would have been mostly or all mixed out, and the reservoir would be pretty well mixed again. All right, let me get my laser pointer back. Okay, so here's just a couple of snapshots in time of the anoxia in Brownlee Reservoir in August of 3 different years. And again, this is dissolved oxygen with red representing anoxia in Brownlee Reservoir. You can see that in these different years that the extent of the anoxia is different. That extent varies year to year, and it's driven by inflowing nutrients in organic material. This is a plot by Jesse Namick in his recent publication. On the x-axis, we have nutrient concentrations coming into Brownlee Reservoir. On the y-axis, we have the volume of anoxic water in Brownlee Reservoir. And this, with the three plots here are chlorophyll A, total phosphorus, and organic nitrogen. So you can see for each of these, we have a really nice relationship where we have, the individual points in these plots are individual years from 2005 to 2021. So when we have years with low nutrients coming into Brownlee, we have a low volume of anoxic water in the reservoir. And when we have years with high concentrations of nutrients coming into Brownlee, we have large volumes of anoxic water in the reservoir. So this inflowing nutrients are really important to determining that volume of water that's anoxic in Brownlee Reservoir, which you'll see is really important to the methylmercury situation. Okay, so the next key point or key finding is that methylmercury is produced and accumulates in the anoxic and hypoxic zones of the water column in Brownlee Reservoir. So the previous slides showed that anoxia development in Brownlee Reservoir through the summer months. Now I want to show you how that anoxia relates to methylmercury. So on the left, I'm going to again show a series of cross sections of dissolved oxygen in Brownlee Reservoir. And again, the scale is blue represents oxygenated water, red is anoxic water. On the right, I have vertical profiles of filter passing methylmercury in the water column at different locations in the reservoir. So the x-axis is filter passing methylmercury concentration. The y-axis is elevation, which you can think of as depth, but we use elevation because the water surface actually fluctuates up and down, so depth gets a little bit confusing. So we use elevation there. The locations that we're talking about here, river mile 300 is about midway through the reservoir. River mile 286 is near the dam. And then I'll show a couple of profiles at river mile 310 as well, which is up in the transition zone. So in April, you see that the reservoir is mostly blue and green in that little plot. That means it's well oxygenated. And over on the right, we see the filter passing methylmercury profiles are pretty much straight up and down, indicating that there's really no difference in concentrations with depth. Homogeneous methylmercury with depth in April. Stepping to May, we see some anoxic conditions developing at depth in the reservoir. We have two different sets of profiles 2 weeks apart in May, and we see maybe a little slight uptick in filter passing methylmercury in the most sample, but these profiles are still pretty much up and down, pretty well mixed. In June, over on the left, we see more anoxia, and it's creeped up into the transition zone here. We have a profile at 310, which shows that anoxic water. We have very clearly elevated concentrations of filter passing methylmercury in that anoxic water. We also see concentrations at 310 in the hypolimnion start to increase. Stepping to the end of July, more anoxic water in the reservoir, and those hypolimnetic methylmercury conditions are continuing to increase at river miles 300 and 286. Moving to August, again, increasing concentrations in the hypolimnion. To September, we collected a lot more samples than the other months, including another sample here at 310 up in the transition zone. And note here that the profile is just straight up and down. There's no more elevated methylmercury at depth at 310 in September. And if you look over on the dissolved oxygen plot, you see that there's no more anoxic water at that location in September either. But at these other locations, we see definitely elevated concentrations in the hypolimnion still. October, still pretty elevated in the hypolimnion. November, our anoxic water is getting a little bit smaller in volume, but we still have elevated concentrations of methylmercury in the metalimnion. And then finally, in December, our profiles are again straight up and down. So those hypolimnetic concentrations are once again similar to what's up in the epilimnion. Again, we don't have dissolved oxygen profiles for December because the sensors froze. But if we did, I think we would see this mostly or completely oxygenated and mixed. So this set of plots, I think, shows pretty nicely this relationship that we see between the anoxic conditions in the water and filter passing methylmercury in the water column. This plot takes that one step further. This is a plot by Ben Peterson that's in a draft manuscript right now. So on the x-axis here, Ben has the redox conditions with oxygenated water, anoxic water in the presence of nitrate, and then moving on to anoxic water in the presence of sulfide at the far side. On the y-axis, we have filter passing methylmercury concentrations. So we have relatively low filter passing methylmercury concentrations when we have oxygenated water, and elevated filter passing methylmercury when we have anoxic water in the presence of nitrate. Moving off to the right, we have the highest methylmercury concentrations in sulfidic conditions, but those are very rare in Brownlee Reservoir. We almost never see sulfidic conditions in Brownlee. So nitrate or manganese and iron reducers are really driving the majority of methylmercury production in Brownlee Reservoir. So I think this has an important management implication in that some places have used nitrate or manganese amendments to control methylmercury production. These data suggest that in this setting, those might not work and may actually be counterproductive. Here's another plot by Ben Peterson, similar x-axis with redox conditions, but the y-axis now is the HGCA abundance. HGCA is a methylation gene. So we see low abundance of this methylation gene in anoxic water or oxygenated waters, excuse me, and increasing abundance of this HGCA gene in anoxic waters in the presence of nitrate and anoxic waters in the presence of sulfide. And then the second plot here on the right is HGCA abundance on the x-axis and filter passing methylmercury concentrations on the y-axis. And you can see there's a pretty nice relationship here. So these results and others from our study suggest that the large portion of methylmercury production in Brownlee Reservoir is happening in the water column. We wouldn't see this relationship if benthic flux of methylmercury were the dominant mercury source. We see very little evidence of significant benthic flux of methylmercury to the water column in Brownlee. We also see enrichment of methylmercury on particles as they move through Brownlee Reservoir, but only during the destratification period, August to December, which is highlighted here in gray. So this is a plot by Brett Poulin, which is in a manuscript that was recently submitted. This is a 2-year time period on the x-axis. The upper plot shows particulate methylmercury concentrations in nanograms per gram, the amount of mercury on particles. And then the lower plot is the percent of particulate mercury as methylmercury. And then the points, the different colors, correspond to different sites. The yellow is at the inflow to Brownlee Reservoir. The white and blue dots are different reservoir locations within the reservoir. And then the green is at the outflow of Brownlee Reservoir. So if we focus on the top plot here and focus on the yellow points, we see that the yellow points are pretty much flatline year round. There's no real seasonal variability. And those concentrations are pretty low, around 5 nanograms per gram, even during this destratification time. Again, those are at the inflow to Brownlee. Within Brownlee and at the outflow of Brownlee, so these blue and purple and green points, we see very clear enrichment of these particles with methylmercury during this destratification period to concentrations of 60 to 100 nanograms per gram. The lower plot here tells essentially the same story, but this is again the percentage of particulate mercury as methylmercury. The yellow points, Brownlee inflow, we see percentages in the 5 to 10 range, fluctuating a little bit, but no clear pattern. Whereas at the outflow sites, we see percentages in the 35 to 45 range. Okay, so and lastly on this topic, anoxia is not only an important control on methylmercury production within Brownlee, it's also important on methylmercury export from Brownlee to downstream. So this plot shows volume of anoxic water in Brownlee on the x-axis and methylmercury concentrations at Brownlee outflow on the y-axis for 7 different years. And we can see that when we have a large volume of anoxic water in Brownlee, we have high concentrations of methylmercury coming out of the reservoir. And the opposite, when we have low volumes of anoxic water in Brownlee, we have low concentrations coming out of the reservoir and going downstream. Okay, so the last point is that fall destratification of Brownlee is a really critical time for methylmercury uptake into the food web and export downstream. Okay, so I think from the previous plots, we've seen that the destratification of Brownlee occurs very slowly from August to December as cool inflowing water plunges and erodes the anoxic zones as shown here. So through this 5-month period, methylmercury that's accumulated in these anoxic zones is mixed into the water column where it's more bioavailable to plankton and other organisms for biological uptake. And we can see that in the data. This is a time series of biweekly samples over a 15-month period, filter passing methylmercury and zooplankton methylmercury. And these are samples collected at the outflows of Brownlee Reservoir and Oxbow Reservoir just downstream. The yellow here highlights the destratification period, and you can see that there's a clear increase in concentrations in water and in zooplankton during this destratification period. And there's a strong correlation between concentrations in water and zooplankton during this destratification period, correlation coefficient of 0.92. So this destratification period, we think, is and it's the mixing of the anoxic water is what's important here, is a critical time for uptake into the base of the food web. If we look at loads of methylmercury coming out of Brownlee, we see that the vast majority is associated with aqueous compartments. So these plots are, again, a 15-month time period on the x-axis and load of methylmercury in grams per day on the y-axis, and as a percent of total load on the plot on the right. We found that the greatest portion of the load was in the filter passing form. Again, this is loads coming out of Brownlee Reservoir. The greatest proportion is in the filter passing form, followed by methylmercury on particles, shown in purple. Only about 5% of the load coming out of Brownlee was biological. And by biological, I mean zooplankton, other invertebrates, fish, and detritus, like leaves and sticks. Even so, even though biological is only about 5%, we expect that this disproportionately impacts methylmercury concentrations in biota downstream, because fish downstream are primarily exposed to methylmercury through their diet, not through filter passing methylmercury in the water. Okay, lastly, I want to point out that while the previous slides have talked about export of methylmercury from Brownlee, I'm not necessarily talking about net export. So if we compare loads of methylmercury coming into Brownlee versus leaving Brownlee, is Brownlee a net sink or a net source of methylmercury? For particulate methylmercury, Brownlee is consistently a net sink. More particulate methylmercury comes into Brownlee than leaves Brownlee, pretty much year-round and year-to-year. This plot shows different years and methylmercury net gain or loss, so the difference between the load coming in versus going out. So a positive bar greater than 0 would indicate a net gain or a source of mercury. A negative bar would indicate a net loss, or that Brownlee is a sink for that mercury. So you can see that particulate methyl is always negative, so Brownlee is always capturing more particulate methylmercury than it is sending downstream. But filter passing methylmercury is kind of a mixed bag. Some years, filter passing methylmercury, or Brownlee is a net source of filter passing methyl. Some years, it's a net sink for filter passing methyl. And again, that variability appears to be related to the volume of anoxic water in the reservoir, which is related to the inflowing nutrient concentrations. So to end this on a positive note, this set of plots is a WRTDS trend analysis by Jesse Namick, which shows trends of nutrients from 1995 to 2020 in the Snake River coming into Brownlee Reservoir. So we see chlorophyll A, total suspended solids, total phosphorus, all have decreasing trends at the inflow to Brownlee Reservoir. So this is good news. Total nitrogen is kind of a no clear trend up or down. But these results provide some hope that with additional work to reduce nutrients and organic matter in the upstream Snake River, for example, through conversions from flood irrigation to sprinkler irrigation, through some of these types of projects, we may be able to reduce anoxia in Brownlee Reservoir and then hopefully lower methylmercury concentrations in the food web as well. So the next steps for this project are Portland State and Reed Harris Environmental are finalizing a C=W2 mercury predictive model. Idaho Power will use that model to test management scenarios. and develop a mercury management plan. Results from this study and from that model will also be used to design and maximize the effectiveness of upstream projects to reduce nutrients and organic matter. Oregon DEQ is working on a Snake River mercury TMDL, which the study team is actively involved with and which will rely entirely on data from this study. And then we have several publications that we expect will be coming out in 2023 on the topics that I've talked about here and several other topics as well. And with that, thank you all for listening, and thanks to all the study team members. And I'll take questions if we have time.
We do have time. Thanks, Austin. That was fantastic. It's a very clear story after 7 years of plus of work on this. So thanks for sharing it. Do you see the chat window?
Let me.
Your colleagues have been peppered with questions and answering them. So thanks to those guys. But you might pick up some of those too.
Let's see.
Maybe who is it, Nick or Brett? You were answering some of the questions. Maybe you could direct Austin to any others.
Yeah, or Brett, feel free to answer a question yourself.
Yeah,
maybe,
I'm happy. Well, I guess first off, if Austin, that was a great presentation. You did a great job representing our group. If I could just verbalize one of the a couple of responses I had to questions. Mark Silos asked about which organisms have the mercury methylation genes. And so, you know, that's a product that we're about to submit to a journal in the next week. Ben Peterson's method for this is really the state of the science for microbial analyses using metagenomic sequencing. And so, you know, what he did in this case is when we were out on the reservoir during collecting those profiles down the water column and getting really fine scale information on the geochemistry of the system, he was collecting DNA samples, and then he sequences all the DNA. He searches for the genes that allow for mercury methylation, and then actually can reconstruct the genomes of the organisms that have the genes. And the power of that method is that you can actually look at the metabolic capabilities of that organism. And so in some cases you can, you know, these organisms some of them can be very restricted in terms of their metabolism, and some of them can be very diverse in terms of what they can use to dump electrons on. And what we've observed is that there's a lot of organisms with the HGCA genes, different organisms within the reservoir complex, and it varies in space and time. But what is consistent is we see the HGCA genes within an organism that can operate on nitrogen, specifically nitrite reduction. So if you if you know a little bit about denitrification, it's a multi-step process, and often organisms are working at different steps in that reduction process. We also see it in fermentative organisms, so organisms that are breaking down large organic molecules. And we saw it in one case in a metal reducing bacteria. We see a lot of manganese reduction within the reservoir. And then lastly, we do see it in the limited cases where we observed sulfate reduction. We saw the genes within sulfate reducers. Now, in terms of the number of samples he analyzed, it was something like maybe 50 total samples to do metagenomics on, which is just enormous. It's more than any other freshwater study to date. We only saw the sulfate reducers in like 2 of the cases. And so the dominant redox conditions that we see is higher on the redox ladder. What we don't know at this point is the relative importance of those various organisms. And that's, you know, that's kind of for the future work. The other question that I could just briefly speak on is Stephen asked about, you know, why we monitor for sulfide within the water column. And the reason for that is that, you know, it's the byproduct of sulfate reduction. And so if we're trying, there's lots of sulfate in the system, and Austin didn't have time to go into that, but there's about 40 ppm sulfate within the reservoir, which is a high sulfate load. A lot of that's from agricultural practices that are up gradient, we believe. And so we're not going to be able to really detect much sulfate reduction looking at just the sulfate concentration. And so we are going to look for the byproduct of dissimilatory sulfate reduction. One of the challenges with sulfur, though, is that the byproduct is reactive. It can reoxidize to sulfate. It can oxidize back to thiosulfate, which is an intermediate, or it could react with it could react with elements like inorganic mercury or other metals and precipitate minerals. And so when we're out on the reservoir, we needed to kind of use a multi-pronged approach to validate that when we don't measure sulfide, that it's really that, sulfate reduction isn't occurring. And so we did that. We use ion selective electrodes that have a pretty low detection limit of 5 ppb. We also measured for thiosulfate, which is that oxidative product. And then what's new for this project is that Ben Peterson is actually able to screen for the genes for sulfate reduction. And consistently what we see is when the genes are present, that's where we observed sulfide accumulate. And when the genes were absent, that's where we didn't observe sulfide. And, simply speaking, if the genes aren't there, the microbes can't do it. And so we're very confident that we've accurately captured the redox state of the reservoir. And so that's kind of how we've approached that.
That's great. That's amazing. You can do all that with the data, a wealth of data.
Yeah, thanks, Brett.
There's a question, chat from Scott McReynolds at DWR. You can address that one.
Yeah, this is a long one. I was trying to
sorry.
read it as Brett was finishing up there. So Scott's asking about, I think, interannual water year differences, like drought conditions, and also HABs, and HAB die-offs driving anoxic conditions at the sediment water interface. So we definitely have HABs in Brownlee, pretty extensively, pretty consistently, I think year to year, but variable from year to year as well.
Yeah, my question is, do you generally see worse problems when you have a wet year and a full reservoir? Because we've had some big blooms in Lake Oroville in California, and it's only when we have a wet year the reservoir fills up, and my hypothesis is that's due to increased nutrient loading. Low water years and drought conditions, we don't get the same amount of nutrients into the lake, so we don't see the HABs, which is kind of counterintuitive. And we are loaded with sediment mercury, and the lake is consumption advised. And I'm just trying to understand if it's kind of a similar process driving it, the nutrient loading, and then how the HAB die-offs when we have big blooms could actually be increasing our methylmercury production. We haven't looked into it very closely.
Yeah. So Jesse, this kind of gets into the paper that he just published. Jesse, do you want to respond to this?
Yeah, I can give a little bit of info about the nutrient loading in high water years, and that's something that, yes, I think we, in the more recent years, during which we had the mercury study ongoing, I think we did see that pattern where 2017 was one of our highest water years on record, and it had the most intense anoxic conditions and the highest amount of methyl accumulated in the anoxic water. I can't say that pattern necessarily held true all back through the 27 years that we have some oxygen data and some nutrient data, but I think some of it may be becoming more clear now that we are seeing declines in inflowing nutrients. I think in the past we had, inflowing chlorophyll A that was, 50 to 100 micrograms per liter all year long, and you just didn't really see the pattern as clearly as now when you have a high water year with, 20 or 30 micrograms per liter chlorophyll coming in. So algae relatively low, but a lot of water, a lot of loading. So I think that pattern is becoming clearer in our data set, and then about the same like you said, where the high loading and high water years are some of our most intense anoxic conditions. Those are the years we would have seen sulfide down deep. So that makes
Awesome.
sense. As far as the HABs situation, boy, we struggle to pick out a pattern, except for bright sun and hot temperatures. HABs love that. But other than that, as far as water years and loading, maybe Nick has some idea. He's had his hands in HABs a little bit more than I have, but I would say they just seem to be pretty consistent every year. We're likely to get HABs, likely in the same spots, and some years are worse than others. And hot sun, hot water and a lot of sun is the one thing that seems to help. And then really, really smoky conditions seem to not help. And so some of the years when we've seen, HABs that aren't as big as we would have expected actually were really, really smoky conditions socked into the canyon.
Yeah, well, Lake Oroville, 70% of the upper watershed burned in the last 5 years. So I'm anticipating much increased sediment and nutrient loading when we have a wet year. So
Sure.
I appreciate the insight. I think that in our system, the HABs and the die-offs could be causing similar spikes in methylation in our lake.
Yes, Scott, I'll just add one thing. We haven't focused on that for Hell's Canyon like Austin and Jesse mentioned, but the USGS lab, the Mercury Research Lab, is starting to look into that for that reason that you mentioned, that anytime you have a lot of deposition in the benthos, you're likely going to exacerbate issues like mercury methylation. And so
Yeah.
Sarah Jansen, who's is taking over for Dave Krabanoff's group, is kind of teamed up with a HABs person at the USGS. So if that's something you're more interested in, she'd be a great contact.
That sounds great. I appreciate it.
Austin, I don't know if you saw the last text from Mark Selos, the philosophical, not so philosophical, but more practical. Like, how'd you guys get all this work done? Was it USGS funded or the what is it called? Idaho Power?
Yeah, both. It was about half and half USGS funding and Idaho Power funding, and the USGS funding coming from a few different pots of money. So, we're basically pooling money from different places, and Idaho Power funding about half of it as well, or a little over half of it. Yeah, so I mean, it is a pretty unique and special study in terms of our, a 10-year study. That's pretty rare for us to be able to work on something for 10 years. Usually it's 2 or 3 years is more common. And being able to pull in, people from all these different disciplines, a lot of stuff that we didn't even touch on today, like the biological work that Colin Eagle-Smith and his group are working on. It's pretty special and rare, and I don't know if I have any great tip to give that, to make it happen again. If I did, I would have another study lined up just like this one to work on for the next 10 years, but unfortunately I don't.
One related thought is, in California, about 15 years ago or so, there was initial efforts to develop in California statewide reservoirs, mercury TMDL, and that one it stalled. So there's not a regulatory driver like the TMDL that this is your work's informing. So that's if that gets resurrected or assigned to staff to work on it, maybe it'll happen here.
Right.
Yeah, and for interest's sake, it is a regulatory driver for Idaho Power that's really driving the study. I mean, we are working on a new federal license, a new FERC license for this complex of hydroelectric facilities, and part of the state's requirements for a 401 water quality certification was to address state standards. One of them is mercury, and very little is known, and so they needed us to propose something to address that, and we said we're going to have to study it. And so that's where we started in, and the idea in their requirements for Idaho Power, they say propose a methylmercury management plan after a certain amount of time, and that's what we're working on. But it's a pretty heavy lift since there's not much known.
Maybe I see there are some comments, but they're sort of some answer there. One last one before we go to close out at the top of the hour is a bit of this. So what? Like how is it going to be used? I guess that's a work in progress right now. The science is informing the future policy, but have you identified any clear management structure activities, things that can be done to reduce the methylmercury in the fish in the reservoirs?
I think it goes back to, trying to control nutrients coming in. If nutrients drive the anoxia, anoxia drives the methylation, then we need to look upstream and try to control those nutrients coming in. And so Idaho Power is already doing some work on that, like sprinkler irrigation or converting flood irrigation to sprinkler irrigation, right, to reduce the amount of sediment and nutrients and organic matter coming into the river. And I think based on our study results, Idaho Power will be starting up some new initiatives as well. Jesse, do you have anything to add to that?
Yeah, I would say that there's promise that could be effective. I mean, there's still a lot of opportunity for those types of projects upstream to make a real difference in inflowing nutrients. The feasibility part of it is a bit of a question mark. I mean, a big question mark right now. I mean, it's a lot of irrigated lands that would need work. So those are, but I think that's kind of where the science is pointing us, is that Anoxic and Brownlee Reservoir is sensitive to what comes in. We have long-term trends. It is an effective strategy to improve anoxia, to improve those conditions. So that's where we're looking at with a lot of energy right now. Once the model is complete, I think that we will be, it would be amiss of us not to focus on some other things that we kind of look at as more Band-Aid things, like potentially in-reservoir actions or water management actions that might help the situation. But I think the big thing that we're looking at is, like Austin said, is continuing to improve that inflow condition to help with anoxia.
Yeah, I think the size of Brownlee, makes some of those in-reservoir Band-Aid type treatments, pretty difficult. Some, stuff that might work on a small reservoir just isn't practical. Not only the size but also the remote location of Brownlee.
Yeah. All right, thanks guys. That's it's, again, great work. You can see some of the appreciation in the chat there. Let me close it off. I just noticed before the webinar today, the Calms web page for webinars is not updated. Didn't list this one, so we'll update that and put a recording to this up there for folks to watch afterwards if they didn't catch it or whatever. And thanks Austin and the rest of the team for this, and everybody else. Look forward to another webinar. We'll start announcing it. There's one, like I said, every month. So the next one's I think March 23rd, something like that. Last week or so in March, and we'll do these through June. So with that, thanks again, and see everybody soon.
Great, thank you everybody.
Thank you.